Hi, everybody. We can start introducing today's webinar, actually the last webinar before our summer break, and also a little bit later than normally in the month since we had the Sweden National Day on the last Friday, which was the first Friday of June. It is my pleasure today to introduce two of our UAC PhD students. As you know, this 2025, we are featuring talks and the research updates by our very own UAC PhD students. And today we have a double feature with Kinga Gulyás from the Department of Chemistry and Athina Eleftheraki from the Department of Cell and Molecular Biology. So we are going to start first with Kinga, then we do a little leg stretcher, and after that we'll continue with Athena before closing off the webinar. Just a couple of housekeeping things. I am recording this webinar. It will be uploaded to our YouTube channel later on. So for the questions, if you don't want your voice or your camera to be on the recording, please write the questions on the chat and I will send them. We will talk to them about them with the speakers. Or if you want, you can also put your hand up during the Q&A to ask the questions yourself directly to the speakers. And without further ado, I would like to introduce Kinga Gulyás, PhD student at the Department of Chemistry at BMC in Uppsala, who is going to talk to us about her latest research paper, which is actually a really good publication in the journal Communications Chemistry. And it's about a new type of beta-lactamase inhibitors, which are not yet in the clinics, which are inhibitors against metallo-β-lactamases. And with that, I will stop sharing my screen and open up the digital stage for you, Kinga, to share your screen.
Thank you very much. I hope you can hear me and soon see my screen.
Yes, I think so. Yes, we can see your screen.
Sorry, now I have a bit of a problem because I don't see it. Sorry.
No problem.
Of course, we tried and it worked. Okay, now I have it.
Perfect. Yes, we see the full screen presentation.
Sorry for this.
Lovely. Looking forward. Thank you, Kinga.
So thank you very much for the kind introduction. And hi, everyone. My name is Kinga, and I'm, as Eva mentioned, a PhD student at the Department of Chemistry in the section of Organic Chemistry, and of course in the Uppsala Antibiotic Center. And my supervisor is Mate Erdei. And my whole PhD project is about the development of novel metallo-β-lactamase inhibitors. And today I'm going to introduce my first big project that is about the development of dynamically chiral phosphonic acid type metallo-β-lactamase inhibitors. As all of us or most of us probably knows, antibiotics have been fundamental for modern medicine. Our healthcare system highly relies on effective antibiotics. Without them, a simple wound infection that is easily treatable nowadays could be deadly again, as it was before basically the discovery of penicillin. Penicillin was called a miracle drug during the Second World War and is estimated to have saved over 200 million of lives up to date. So we still take antibiotics granted and we still have a couple of working classes, but they are less and less effective. And a very, very important and big class of antibiotics is the beta-lactam antibiotics. They are very successful, although they have been more or less the first one being discovered. And penicillin is part of this group. They are cheap, they are available in the low and middle income countries. They have a broad spectrum activity against many gram-positive and gram-negative pathogens, and they have a very kind of nice low toxicity profile. This is also acknowledged by the WHO that listed or published the model list of essential medicines. And here you see the group of essential antibiotics, and half of the essential antibiotics come from the single group of beta-lactam antibiotics. Unfortunately, resistance occurs against this group of antibiotics as well, which is again highlighted by the WHO, who published the bacterial priority pathogens list, which is a list of 12 pathogen families that pose the greatest threat to human health. And in total, 7 out of the 12 families, pathogen families, and all the pathogens out of the most critical group are resistant to beta-lactam antibiotics. And this highlights very well that we need to find a solution for the resistance problem against this group of very essential beta-lactam antibiotics. The most clinically relevant and most widely spread resistance mechanism against this class of antibiotics is the expression of enzymes, the so-called beta-lactamases by bacteria that are able to dismantle the antibiotics. So there is this 4-membered beta-lactam ring that is essential for all beta-lactam type antibiotics. It is important for their activity, and these enzymes are able to open this ring, and the open form of the antibiotics is not active anymore. There are 2 types of these enzymes, the serine and metallo-β-lactamases. There is thankfully a potential solution to this problem, which is combination therapy when you administer the existing drug together with an enzyme inhibitor. And then the inhibitor basically occupies the enzyme, and the antibiotic can act as before this resistance occurred. And this works and is on the market from the 1980s. For example, Augmentin is a combination drug, which is among the most selling antibiotics worldwide. However, it only works so far against one group, the serine beta-lactamases, and there is no inhibitor in the market against metallo-β-lactamases. Metallo-β-lactamases do pose the big problem nowadays. They basically are able to open all classes of this beta-lactam antibiotics. And yeah, there is no solution at the moment against these enzymes. Yeah, there are a few promising inhibitors in clinical trials, and they have this typical bicyclic boronate structure. However, they haven't reached the market. There is already resistance development against them. So, as I mentioned, metallo-β-lactamases cause the biggest problem nowadays, and they are the clinically most relevant. So we selected our target enzymes from this group. We have 3 targets at the moment: NDM1, VIM2, and GIM1. NDM1 and VIM2 are widely spread over the world, and they cause a lot of deaths. And GIM1 is another enzyme that has the most different binding pocket among this enzyme family. Here on the first picture, you can see the overlay of the secondary structure of these 3 enzymes. They are all similar in size, around 230 kilodalton, 250 amino acids. They all share this alpha, beta, beta, alpha fold, but the overall sequence similarity is only around 25%. So it has been proven very difficult to find an inhibitor that is broad spectrum and can bind into the binding pocket of all of these or many of the members of this group. They all utilize very flexible loops, so they have a very broad substrate profile. When you want to target different enzymes within a family, you need to rely on things that are common in all of these enzymes, and we cannot really rely on the sequence similarity, but there are a few things that are common for all these enzymes. And one of this is that all of them have 2 zinc ions in the binding pocket, which is crucial for their activity. And the other one is that the hydrolysis mechanism that they use to open up the beta-lactam ring is the same, and is proposed to proceed via tetrahedral transition state. So in this way, we can design transition state analogs that mimic this tetrahedral geometry to have a common binder. And these are two key considerations that we use in our inhibitor design. So we utilize phosphorus as our key element because phosphorus is known to have this tetrahedral geometry. So it's possible that it can mimic this tetrahedral transition state. And phosphonic acids are known to have affinity to metal ions, so they can bind to the zinc ions in the active site. Our inhibitor is further inspired by existing, for example, serine beta-lactamases like vaborbactam, potential metallo-β-lactamases like the cyclic boronate that I have shown before, and also existing beta-lactam antibiotics. As metallo-β-lactamases cause a lot of problems in gram-negative bacteria. When you design an inhibitor against them, you have to keep in mind that they have to cross the gram-negative outer membrane. So in our design, we try to balance the water solubility and the lipophilicity of these compounds with having a hydrophilic phosphonic acid core together with more lipophilic thiophene, benzothiophene, phenyl, and benzyl side chain. To withstand the rapid resistance development, one has to come up with completely new structure, and then for being able to adapt to the structural differences in the different enzymes, we incorporated dynamic chirality in our compounds. So we have a stereocenter in a benzylic position next to a phosphonic acid and an amide moieties. And this position is ought to be easily deprotonable under physiological conditions, enabling the interconversion of the two stereoisomers. So the synthesis was very straightforward. It goes through 4 steps, which starts with the Kabachnik-Fields reaction when we get the key alpha aminophosphonate structure. Then the amine functionality is deprotected, where we get the free amine that enables easy coupling of virtually infinite amount of carboxylic acid to get the final core structure. And the last step is only the cleavage of the methyl ether and then the ester hydrolysis in one single step. So I synthesized 13 compound, and then we needed to evaluate if our design was successful and if they bind to the target enzymes. So first we conducted IC50 measurements against the purified 3 target metallo-β-lactamases, and thankfully all the compounds showed some activity. The best ones showed low micromolar activity against at least one of the enzymes, and which was nice that we saw that some of the inhibitors even showed activity against 2 or even 3 of the enzymes in the low micromolar range, which is promising when you actually aim for developing a broad spectrum inhibitor. We also measured their cytotoxicity against HepG2 cells, and they were thankfully not cytotoxic. But as I mentioned, they have to be able to cross the gram-negative outer membrane. So we also conducted membrane permeability experiments, which is on figure B. Here you see the bars representing the 3 different enzymes with different colors, and the bars show the relative activity of the enzymes. So when we don't add the inhibitor, we just add the buffer, the relative activity of the enzymes is 1, so they are fully active. We have a control captopril, which decreases the relative activity of the enzymes. And what you can see here that basically all of our enzymes decrease the relative, all of our inhibitors decrease the relative activity of the enzymes. That means that the inhibitors are able to go through the gram-negative membrane and have some activity. Then lastly, we also evaluated some of our compounds with real clinical isolates, and then we saw up to 30% zone inhibition against Vim2 expressing clinical isolates. So after confirming that they were active, we wanted to actually see how they bind, where they bind in the enzymes, and how differently do the different structures bind. And one problem with this metallo-β-lactamase is that it's different to crystallize them. And even if once you got a crystal with one inhibitor, it's not guaranteed that if you have another inhibitor structure, you will get a crystal again. And one potential solution is to study binding in solution close to physiological condition with solution NMR. And for this, you need the enzyme expressed labeled, so it's NMR active, and then you have to do the backbone assignment in solution. And thankfully, the backbone assignment for Vim2 and NDM1, two of my target enzymes, have been done before in our group. And here you see proton nitrogen HSQC spectra of the two enzymes, where each peak basically means one amino acid in the enzyme backbone. So you have to identify, like, yeah, this peak is, I don't know, serine 32. And when you have this in hand, you can study any kind of inhibitor in solution. So with the backbone assignments in hand, I conducted NMR titration experiments with Vim2 and NDM1. And here you see my results from Vim2 bound to these 5 of the inhibitors that I had. The gray peaks represent the free enzymes when there is no inhibitor added. And then I add increasing amounts of the inhibitors to the NMR tube. And then if there is any interaction, you see that these original peaks will move somewhere else. If there is no binding or no effect, then they will just overlap. So the enzyme is not going to be affected. So it's going to be the original position of the peak. And here with colors, you see the overlay of the 5 different inhibitors. And what you can see here quite nicely, that all these colored peaks kind of overlap. And then the magnitude of their movement is also very similar. That indicates that all of these compounds occupy the same binding pocket. And then it also indicates that they have similar affinity to the enzyme. This is an example how I determine where the inhibitor binds. So with blue is the free enzymes, and with green it's the most potent inhibitor titrated. And then as I knew which peak corresponds to which amino acid, I could indicate that which amino acids were affected by the binding. And here is the binding pocket that was determined by solution NMR. And what we saw that all these amino acids that are affected by binding are in flexible loops 3, 5, 10, and 12, and also amino acid residues around the zinc ion, which was our goal because with the phosphonic acid, we wanted to target the zinc ions. So I did NMR binding studies for Vim2 and NDM1, and this is just a representation with the most potent inhibitor of the two enzymes. But you can see that the loops affected are the same both for NDM1 and Vim2, and also that the position and the binding mode is very similar for the two different enzymes. And thankfully, we also got a few co-crystals with Vim2, which further confirmed us that the inhibitors bind in the binding pocket that was determined by NMR. And the key interaction is between the phosphonic acid core and the two zinc ions. But what was new and we saw in the co-crystal is that both enantiomers do bind in the same binding pocket with a 180 degrees flipped binding mode, which is quite a new thing in inhibitor design because usually only one steroid isomer binds in the binding pocket. And that meant that Vim2 is tolerant to a rare scale rotation in the active site. It was too demanding to do NMR studies or get crystals with all the 13 inhibitor and 3 enzyme combinations. So we developed and validated a computational docking method starting from or based on our co-crystal structure. And then we docked all of our inhibitors to all of our enzymes. And the conclusion was what we saw previously, that the key interaction is always between the two zinc ions and the phosphonic acid core, regardless of which enzyme it is. And it's these compounds always bind in the same binding pocket of all the three enzymes. And the affinity of the two stereoisomers is always very similar, but the binding mode is always flipped by 180 degrees. So this basically indicates that we did get or we designed a broad, hopefully potentially broad spectrum inhibitor. And our incorporation of dynamic chirality wanted to withstand, first of all, the rapid resistance development, and also was designed to have broad spectrum activity. And then to see if it could potentially withstand rapid resistance development, we did saturation mutagenesis, and then we docked the inhibitors to these mutated variants of the enzymes. Here you can see that we got around 350, 80 mutants, and then we wanted to see when is the binding weakened. So the idea is that we have the two stereoisomers in solution, and then if the enzyme mutates, it probably weakens the binding of one of the stereoisomers, but that's not a problem because the other stereoisomer still binds, and we have this dynamic interconversion between the two stereoisomers. So even if one is weakened, it can still convert to the stronger stereoisomer, and then we still have binding. But the problem arises when the binding is weakened for both of the stereoisomers, and then we wanted to see in how many cases this happens. And you see here that the worst case is for this compound 5C, when both stereoisomers gets weakened, their binding gets weakened, then it's more or less 10% of all the mutations, but this was the maximum we got. So, for example, for Vim2, even if there are very few mutations when both of the stereoisomers binding affinity gets weakened. So this indicates that this could be a potential strategy to withstand rapid resistance development. To reflect and conclude this first project I had, we developed a new phosphonic acid type series, and we successfully incorporated dynamic chirality, which could be maybe a potential solution to withstand rapid resistance development and have a broad spectrum inhibitor. We did binding studies by solution NMR and X-ray and computational methods, and then the overall aim of my PhD is to further improve these initial inhibitor candidates to have better and stronger binders. And of course, I would like to thank a lot of people. First of all, my supervisor and my group, and all our collaborators who either helped us doing some experiments or taught me how to do the experiments and helped me doing these experiments, and all the infrastructure we have at Uppsala, and a big thank you for the Uppsala Antibiotic Center, and thank you for your attention.
Thank you so much, Kinga. You are getting some applauses also for me. It's really nice to see it all, completed. As so far as a project can be completed. Obviously, there can be some follow-ups, and maybe we can talk a little bit about there. We have some minutes for discussion. First of all, I just wanted to say I read the paper, and now when you explain this NMR titration experiment is so much more clear to me, how
Yes,
it actually works.
I try to adjust my talk to the audience because I know not everyone is familiar with this
Yeah, so it's really nice. I wonder, within the development of this project, what has been the most challenging part on your side to get this going and to get to the point where you have it now?
Hmm. I think the NMR titration studies, because this is also a new thing for me, and it's a lot of peaks in the spectrum, and you really have to be careful, like which peaks moves where, and like not to mess and mix up.
Do you
Um,
do that by eye? Is that
yeah,
done by eye? You basically look at each one of them and then you try to see where did they move to and try to.
yes. So
Oh,
I do have a series of spectra that I overlay. So I start with like 0.25 equivalent of inhibitor and then 0.5, 0.75, 1. So I have a lot of them so I can follow the gradual movement of them.
so is that the purpose of doing a titration of the amount of it? Is that
Yes, exactly.
so? Is
Because
that
if you
ah?
to be able to follow, because if you would just have the starting spectrum and the end one, you don't know which peaks moves where. Any of them could move anywhere. So you want to see the way how they move, and like
This
it's
is so
really
cool. This is even
interesting.
more clear now to understand. Yeah, because obviously you are presenting it now here, and we believe you. You know that this blue point corresponds to that one.
Yes.
But so that's what the titration
Yeah, exactly.
power
Yes.
comes from that you do it. That's really cool. Um, because I am a biologist, I tend more to be very
Yes.
interested about the biology behind this and the next steps regarding in vivo experiments. So you were talking about simulations, right, of these mutations and these changes, and
Yes.
then trying to see how the change in in the structure will be solved. Well, but it wouldn't be affected by these changes or somewhat like that.
Yes.
But do you have in mind to do this actually in vivo? Is there a way forward there?
Yeah, it would be really cool, but I mean, we need collaborators for that uh,
Mhm.
and resources. So we are not gonna do this at the moment because I mean, I don't have that much time left.
Mhm.
So I started focusing on the development of stronger inhibitors to have lower micromolar or nanomolar, maybe, I don't know, activity. And then if I get um stronger inhibition, then I think we could go further.
Okay, so what you're telling us here is that even though these prove the concept, that these
Yes,
dynamically chiral kind of structures could work, you think that in order to then be developed as a potential inhibitor that can be given with a drug, they need to be even more potent than the numbers that you're seeing here.
yes. I mean, I think we could still work with this to have more experiments and more tests and more studies, but we had to prioritize things for my last year, and then we prioritized getting better inhibitors. Yeah, but overall, yes, we would definitely need to, and I actually would love to check this with experiments. And yeah,
With collaborators as well, yeah, of course.
yes.
By the way, anybody out there here, you are also welcome to ask questions. So when you're talking about developing stronger ones, are they going to be also based on these ones in particular, or are there other ones that you are exploring?
No, I am. It's based on these compounds. Also, we exactly know how they bind now, so we can even target specific positions in the binding pocket. Uh, yeah, so all of them are based on these ones.
How do you decide, once you know the structure, right, the binding of these, um, what is your rationale to decide to change one or another in order to increase their activity?
Yeah, so a lot of rational design comes from experience from literature and also like crystal structures. As I have my own crystal structure, the main thing is that the phosphonic acid core is key. It's very important. It's close to the zinc ions. Um, and then as you, I hope you understand that like basically whatever the other part of the molecule was, it could be even flipped. So
Yeah.
there is no super strong interaction between the amino acid residues and the other parts of the molecule. So I basically look at the crystal structure and the compound in the binding pocket, and I check what kind of amino acids are around it and what kind of interaction those amino acids could give. And I try to estimate in which position what kind of group should be to have that interaction.
Hmm.
And it's all nice in theory, but sometimes when you actually design that compound, it just moves a little bit in the binding pocket, so the distance is not ideal anymore. But yeah, we do something like this.
And you have to try them out and then see.
Yeah, and also whatever idea I have, I check with our computational docking method that we developed, and that's, I mean, without experiments, it's it doesn't say too much, but it's a good starting point.
Mhm.
So computationally evaluate.
So cool. Very great. I've this is a few times I feel like now I understand a little bit more of chemistry, which is really cool.
I hope so. That was my aim.
Yes, it was very clear, and I'm really looking forward to see, the last year how many more you can work with and what you can find. And we
Me
really
too.
do need some of these, maybe in the future, whatever is already on the pipeline that comes out, because we do need to have inhibitors for these metallo-lactamases, metallo-beta-lactamases, which are quite dangerous and something to take care of. If nobody has any questions for Kinga about
No.
this work, just to mention that we are covering this paper also on the podcast episode that will come up on Monday. And we will have there also links to the paper if you wanna, look at it more